The paper introduces RNACG, a universal RNA sequence conditional generation model based on flow matching. It addresses sequence design issues by characterizing them as conditional generation tasks. RNACG accommodates various conditional inputs and is portable, enabling customization for specific requirements. The model is evaluated on tasks including RNA 3D and 2D structure inverse folding, family-specific sequence generation, and 5' UTR translation efficiency prediction, demonstrating superior or competitive performance compared to other methods. This framework offers a novel approach to RNA sequence design and potential methods for simulation experiments with large-scale RNA sequence data. In the realm of computational fluid dynamics, simulating turbulent flows remains a significant challenge due to the intricate interactions between velocity and pressure fields. To address this, researchers have proposed the use of artificial neural networks, ANNs, to approximate the Reynolds stress tensor, a crucial component in modeling turbulent flows. The Reynolds stress tensor, denoted by tau, represents the correlation between velocity fluctuations and is a key component in the Reynolds-Savarage-Navier-Stokes (RANS) equations. However, accurately modeling tau is a daunting task, especially in complex geometries and at high Reynolds numbers. To overcome this limitation, the authors employ a novel approach, leveraging the capabilities of ANNs to learn the underlying patterns in tau. By training a neural network on a dataset of direct numerical simulation (DNS) results, the authors demonstrate the ability to accurately predict tau in a range of turbulent flow regimes. The proposed methodology involves a deep neural network architecture, comprising multiple hidden layers and an outputs layer that yields the Reynolds stress tensor components. The network is trained using a mean squared error loss function, ensuring accurate predictions of tau. Notably, the author's approach is shown to outperform traditional turbulence models, such as the K-omega and K-epsilon models, in terms of predictive accuracy. Furthermore, the ANN-based approach is demonstrated to be computationally efficient, making it a promising tool for large-scale simulations. The results are visualized through a series of figures, showcasing the excellent agreement between predicted and DNS computed tau components. Additionally, the authors provide a detailed analysis of the neural network's performance, highlighting the importance of hyperparameter tuning and dataset quality. In conclusion, this study presents a groundbreaking approach to modeling turbulent flows, leveraging the capabilities of artificial neural networks to accurately predict the Reynolds stress tensor. The proposed methodology has significant implications for the development of more accurate and efficient turbulence models, paving the way for improved simulations in a range of engineering and scientific applications. The RNA design problem is formulated as a function f that maps a given property C to the set of all RNA sequences S that possess this property. Typically, finding the exact sequence S that satisfies the property C is intractable. Instead, we define the function f as finding the sequence S with a property C that closely approximates the target property C within a tolerance epsilon, based on the distance metric d. The optimization goal of RNA design problems is to minimize the expectation of the distance metric d over the target distribution sigma, c. To achieve this, we propose RNACG, a model trained to minimize the difference between the generated sequence distribution and the target distribution. RNACG consists of three primary components: a random sampler with its corresponding embedding layer, a multimodal diffusion transformer, MMDIT, and a downstream linear layer for sequence generation. In conditional generation tasks, an additional condition encoder module is incorporated, customized to process input conditions. The DIC component is a function that takes input x, time t, and condition c, outputting the predicted vector field. This vector field is obtained by applying a linear layer to the DIT output. By leveraging these components, RNACG effectively tackles the RNA design problem, generating sequences that closely match the target property C. The RNACG workflow is centered around the sampling and training strategy, which involves sampling the initial sequence probability distribution from a Dirichlet distribution due to the nature of RNA sequence data. This approach is more suitable for modeling categorical data. The loss function used is cross-entropy loss, defined as the negative log likelihood of the predicted probabilities. The workflow is further evaluated through an experiment on RNA 3D structure inverse folding, comparing RNACG with state-of-the-art methods like RDesign, UniIF, and PyFold. The results demonstrate that RNACG achieves similar performance to RDesign in terms of sequence recovery and MACRIF1 score across different sequence lengths. The authors present the performance metrics of various RNA 3D inverse folding methods, including PyFold, RDesign, UniIF, and RNACG, in Table 1. The results show that RNACG outperforms other methods in terms of F1 score. To further evaluate the performance of RNACG, the authors generated sequences with randomly initialized X0 and reported the performance metrics in Table 2. The results demonstrate that RNACG achieves competitive performance in terms of F1 score and recovery rate. To verify that RNACG has learned semantic information of RNA sequences, the authors generated 20 sequences for each structure in the test set with different mutation ratios and used RNACG to generate sequences for each structure 20 times. They predicted the structures of all sequences using row fold and calculated the RMSD and TM score between the predicted structures and the target structures. The results in Table 3 show a significant difference between RNACG and random sampling, indicating that RNACG has learned local sequence information in addition to global information. The authors then discuss the inverse folding task on RNA 2D structure, which is defined as predicting or generating RNA sequences that fold into target 2D structures without node information. The 2D structure is represented as a graph, where nodes are nucleotides and edges are phosphodiester bonds or base pairs. The authors use a simple MPNN as a condition net to encode the 2D structure information, where edge features are different types of interactions and node features are all initiated as one. The authors define the target 2D structure as G equals empty set E, and the initial input as X, and the inverse folding task is to predict or generate RNA sequences that fold into the target 2D structure. They use a post-processing method to generate the sequence, which is defined in algorithm 1. The method takes the predicted probabilities of each nucleotide and generates the sequence by iteratively selecting the nucleotide with the highest probability. To validate the generated sequences, the authors use NotFold to predict the 2D structures of the generated sequences and compare them with the target 2D structures. They report 3 groups of sequences and 2D structures: predicted 2D structures of generated sequences, predicted 2D structures of true sequences, and true 2D structures of true sequences. The authors present a comprehensive evaluation of RNACG, a novel method for RNA 2D inverse folding, comparing its performance to established tools RNA-inverse and NotFold. Results show that RNACG outperforms RNA-inverse on long sequences, but underperforms on short sequences. Notably, both methods' performance decreases with increasing sequence length, with RNA-inverse's performance declining more rapidly. In addition to inverse folding, the authors address the challenging task of RNA family-specific sequence generation, where the goal is to generate sequences that belong to a specific RNA family. This task is hindered by the high sequence diversity within families and the complexity of covariance models, CMs, used to represent RNA families. To tackle this, the authors employ a model setup that encodes 2D structure information from RNA families represented as CMs. Performance evaluation on the RFAM database reveals that RFAM-Gen and CGVA outperform RNACG in terms of robustness and scores, but RNACG can generate sequences with higher scores, although some may have negative scores. This disparity is attributed to the distinct encoding of family-specific information, with RNACG utilizing 2D structure information and other methods relying on CMs. Nevertheless, RNACG outperforms GVA and CVA, demonstrating its potential for RNA family-specific sequence generation tasks. Statistical analysis using p-values highlights significant differences between RNACG and other RNA sampling methods. A summary table and figure illustrating the F1 score of RNA 2D inverse folding tasks between RNACG and RNA-inverse provide a comprehensive overview of the results. The authors applied their RNACG model to the 5' UTR translation efficiency prediction task, leveraging pre-training on the UTR-LM dataset through masked language modeling and mini-free energy prediction tasks. The results, as shown in Table 4, demonstrate the superior performance of RNACG compared to the state-of-the-art language model UTR-LM across all cell types, characterized by lower root mean squared error and higher Pearson R values. This suggests the potential of guided sequence generation for specified 5' UTR translation efficiency. Notably, the authors acknowledge a key limitation. During testing, true values were used instead of sampling, ensuring a one-to-one correspondence between output and true values. In contrast, real generation processes involve input values as continuous probability distributions rather than one-hot encoding of real values. To address this limitation, the authors propose using accurate prediction of output values for explicit iterative optimization of sequences. The RNACG model is demonstrated to be a universal method for RNA sequence design, achieving optimal performance across multiple metrics. Its potential for direct end-to-end iterative optimization of generated sequences based on outputs is highlighted. However, the authors also discuss the limitations of the model, including the difficulty in determining the sampling space of RNA sequences and the bias introduced by the prior used in the model. Ultimately, the authors envision RNACG contributing to endeavors such as vaccine development and genetic component engineering, pending robust validation in biological experiments. The authors present a comprehensive review of recent advancements in RNA and protein sequence design using deep learning-based approaches. These advancements have far-reaching implications for fields such as synthetic biology and biomedicine. Techniques like generative diffusion models, geometric deep learning, and flow matching are employed in inverse folding RNA and protein sequences. Notable studies include protein MPNN for robust protein sequence design, PiFold for efficient and effective protein inverse folding, and UniIF for unified molecule inverse folding. Ribodiffusion uses generative diffusion models for tertiary structure-based RNA inverse folding, while Grenade employs geometric deep learning for 3D RNA inverse design. RNA secondary structure prediction is also crucial, with studies utilizing deep learning with thermodynamic integration. The RFAM14 database expanded coverage of metagenomic, viral, and microRNA families. E2eFold 3D provides accurate de novo RNA 3D structure prediction. Dirichlet flow matching is used for DNA sequence design, and RDesign offers hierarchical data-efficient representation learning for tertiary structure-based RNA design. Advancements in scalable diffusion models with transformers, partial RNA design, and algorithmic improvements for optimized mRNA design are also highlighted. These studies and techniques are deeply rooted in mathematical and computational concepts, including machine learning, deep learning, and statistical mechanics.